For almost everything that has ever lived on this planet, every leaf, every bacterium, every animal, every human who has ever drawn breath, the story begins in the same place, not in a forest, not in an ocean, but in a single ancestral lineage so ancient that it predates memory, language, even bones themselves. Every living thing on Earth is related, not metaphorically, literally. Trace the chain of inheritance far enough back, and it converges into one shared origin, one deep biological beginning. This is not philosophy, it is genetics, it is chemistry, it is written into every cell of your body. And yet, the scale of this story defies the human mind. Human history stretches back a few thousand years. Civilizations, perhaps 10,000. Our species, a few 100,000 at most. But life on Earth has been unfolding for nearly 4 billion years, a span so vast that it compresses empires into instants and reduces entire ages to thin lines in stone. To understand life, we must abandon intuition. We must trade clocks for geology, memory for molecules. Because life did not arrive suddenly, nor was it inevitable. For most of the universe's existence, there was no life at all. For billions of years after the universe itself began, the Earth did not yet exist. And for hundreds of millions of years after Earth formed, it was hostile to anything that dared to endure. So the question is not simply how life evolved. The deeper question is this: why did life arise here, on this particular world? And why did it follow this path through extinction, transformation, and fragile persistence rather than any other? What you are about to witness is not a celebration of nature, nor a catalogue of species. It is a reconstruction drawn from rocks, isotopes, fossils, and genes of how a dead planet slowly became alive. Before life existed, Earth was something else entirely. And that world is where this story truly begins. Earth did not begin as a cradle, it began as debris. 4.5 billion years ago, long before oceans or skies or continents, the region of space that would one day become our world was nothing more than a chaotic disk of dust and rock circling a young, violent sun. Countless fragments collided, merged, shattered, and merged again. There was no intention in this process, no design, only gravity, time, and chance. Slowly, inexorably, these collisions built something larger, a growing planetary body that fed on everything in its path. With each impact, energy was released. Heat accumulated. The early Earth became a sphere of molten stone, its surface an ocean of magma, its interior churning and unstable. This was not a world that could host life. It was a world still deciding what it would become. As Earth grew, gravity pulled heavier elements inward. Iron and nickel sank toward the center, forming a dense core. Lighter materials rose, creating a mantle and a primitive crust. This internal separation, planetary differentiation, was not cosmetic. It would determine Earth's future. The liquid outer core began to move, generating a magnetic field that wrapped the planet in an invisible shield. Without it, the sun's radiation would have stripped the atmosphere away, as it did on Mars. But even with protection, the surface remained lethal. The young Earth was battered relentlessly. For hundreds of millions of years, asteroids and protoplanets slammed into it in what is known as the heavy bombardment period. Some impacts were large enough to melt the crust entirely. Any early surface stability was erased again and again. If life had appeared during this time, it would not have lasted. The sky offered no refuge. There was no oxygen to breathe. In fact, oxygen would have been deadly to anything alive at this stage. The atmosphere was thick with volcanic gases, carbon dioxide, methane, ammonia, sulfur compounds. Lightning tore through the clouds. Acid rain fell onto incandescent rock. The air itself was poisonous. This was Earth's natural state, violent, unstable, hostile. And yet within this chaos, the foundations of habitability were quietly assembling. Volcanoes, while destructive, were also creative. They released gases that thickened the atmosphere, trapping heat and preventing the planet from freezing. As Earth slowly cooled, patches of solid crust began to persist for longer periods. The surface was no longer uniformly molten. Cracks formed. Plates began to move. A dynamic planet emerged, one that recycled its surface, regulated its temperature, and evolved over time. Cooling did not happen all at once. It happened unevenly, catastrophically, and repeatedly. But with each cycle, Earth edged closer to stability. Deep within its interior, radioactive elements decayed, providing a long-term heat source. This kept the mantle partially molten, driving plate tectonics, a process that would later regulate atmospheric carbon dioxide and stabilize climate across geological timescales. No other known planet does this in quite the same way. Still, none of this guaranteed life. Earth could easily have become a sterile rock, too hot, too dry, too unstable. Many planets likely did. What makes Earth unusual is not that it formed, but that it did not settle into equilibrium too quickly. It remained restless, active, geologically alive. Then, gradually, something extraordinary began to happen. As temperatures dropped below critical thresholds, water vapor in the atmosphere condensed. Rain fell, not gently but violently. For thousands, perhaps millions of years, the surface collected liquid water, basins filled, lowlands flooded. The first oceans began to take shape. This should not have been possible. Given the intensity of early impacts and the sun's youthful volatility, liquid water should have been stripped away or boiled off repeatedly. And yet, geological evidence locked inside ancient zircon crystals suggests that by as early as 4.4 billion years ago, stable bodies of water already existed. Somehow, Earth held on to its water. Whether it arrived embedded in asteroids, emerged from the planet's interior, or both, the result was the same, a global network of oceans covering much of the surface. These waters were not blue and calm. They were dark, mineral-rich, acidic, and hot. Hydrothermal systems churned at the seafloor, pumping energy and chemistry into the depths. The planet was still hostile, but it was no longer uniform. For the first time, Earth contained gradients, hot and cold, acidic and alkaline, light and dark. These contrasts mattered. They created pockets where chemistry could behave differently, where complexity could emerge and persist instead of being erased. The atmosphere, too, began to change. Still oxygen-free, but thicker and more stable, it moderated temperature extremes. The magnetic field deflected solar wind. The crust thickened. Impacts became less frequent. The planet stopped resetting itself completely. This was not the birth of life. It was something more subtle and perhaps more important. It was the birth of opportunity. Earth had transformed from a molten sphere into a chemically diverse, energy-rich world with liquid water, protection from space, and long-term geological stability. None of these elements alone create life. Together, they make life possible. And crucially, this transformation was not inevitable. It was contingent. A slightly weaker magnetic field, a slightly smaller planet, a different orbital history, and Earth would have remained barren. Instead, it became a place where matter could linger, interact, and change, where chemistry could begin to remember itself. What emerged next did not crawl or swim or breathe. It did something far more radical. It learned how to endure. Water was never guaranteed. By every reasonable expectation, a planet forming this close to a young, violent sun should have been dry. Its light elements stripped away, its surface baked and barren, and yet Earth became a world defined not by stone, but by liquid. The origin of that water remains one of the most quietly contested questions in planetary science. Two explanations dominate, neither complete on its own. One suggests delivery from elsewhere, water locked inside asteroids and carbon-rich meteorites that struck Earth during its early history. These bodies, remnants of the outer solar system, carried hydrated minerals and ice. Their isotopic fingerprints closely resemble the hydrogen ratios found in Earth's oceans today, lending weight to the idea that much of our water arrived from space. The other explanation looks inward. As the planet differentiated and cooled, vast quantities of water vapor were released from Earth's interior through volcanic degassing. Steam accumulated in the atmosphere, condensing as temperatures fell. Rain followed, not for days or centuries, but for spans of time that defy human imagination. The truth is likely a combination of both. Water delivered, water released, water retained. What matters is not the route it took, but the fact that it stayed. Evidence for this endurance is etched into some of the oldest materials ever found on Earth, microscopic zircon crystals embedded in ancient Australian rocks. These crystals, over 4.4 billion years old, formed in the presence of liquid water. Their chemistry tells a remarkable story, that oceans may have existed only 100 million years after Earth's formation, far earlier than once believed. This revelation rewrote the timeline. Earth did not wait patiently to become wet. It became wet almost immediately. But these early oceans were nothing like those we know today. They were warm, perhaps even hot, acidic, saturated with dissolved metals leached from a restless crust. The sky above them was thick with carbon dioxide and methane, filtering sunlight into a dim reddish glow. The sun itself burned fainter than it does now, yet the planet did not freeze. A paradox explained by powerful greenhouse gases trapping heat at the surface. The oceans covered most of the planet, creating a continuous global system. There were no continents as we recognize them, only volcanic islands and submerged plateaus. The boundary between sea and rock was everywhere, and at that boundary, chemistry accelerated. Water is not a passive substance. It dissolves, transports, concentrates, and reacts. In Earth's early oceans, it became a medium where atoms could meet repeatedly, rearranging into increasingly complex forms. Carbon, hydrogen, nitrogen, sulfur, phosphorus, the elements essential to life, circulated freely, driven by heat and motion. The oceans did more than hold chemistry, they organized it. At the seafloor, hydrothermal vents punctured the crust, releasing mineral-rich fluids heated deep within the planet. These vents created sharp gradients, hot against cold, alkaline against acidic, reduced chemicals meeting oxidized seawater. Energy flowed naturally across these boundaries. Such gradients are not incidental, they are fundamental. Life at its core exploits differences between inside and outside, before and after, here and there. Long before cells existed, the ocean floor offered environments where chemistry could be driven forward without direction, only persistence. Mineral surfaces added another layer of structure. Clays and iron-sulfur compounds provided scaffolds where molecules could concentrate instead of drifting apart. Reactions that would be vanishingly rare in open water became statistically inevitable when confined to microscopic surfaces. Above, tides rose and fell under the influence of a moon far closer than it is today. Shorelines, where they existed, experienced cycles of wetting and drying. Pools concentrated organic molecules as water evaporated, then redistributed them when tides returned. Repetition favored stability. Stability favored complexity. None of this required intention. It required time, and the oceans had it. For hundreds of millions of years, Earth's seas acted as vast chemical laboratories, not experimenting, not searching, but allowing outcomes to persist. Most molecular arrangements failed immediately. Some lasted a little longer. A few endured long enough to interact again. The key transformation was not complexity, but continuity. Chemistry on land resets easily. Fire, impact, erosion erase progress. In the oceans, continuity was protected. Water buffered temperature. It absorbed radiation. It softened catastrophe. Even massive impacts could sterilize the surface while leaving deep waters relatively undisturbed. This protection mattered, because at some point molecules began to do something profoundly unfamiliar. They stopped merely reacting and began persisting. Certain arrangements could maintain themselves by drawing on energy gradients around them. Others could produce copies, imperfect but recognizable, of their own structure. These systems were still chemistry governed by physical law, but they introduced a new property into the universe: memory. Not memory as thought, but memory as structure. The ability of matter to carry information forward in time. The oceans did not create this transition deliberately. They did not guide it. They simply made it possible by refusing to erase it. This is why the oceans matter more than any single molecule or reaction. They were not just environments, they were stabilizers. They allowed fragile processes to repeat long enough for selection, blind and mechanical, to begin favoring what lasted. And once something can last, it can change. Once it can change, it can compete. Once it can compete, the rules of the planet shift permanently. The oceans that should not have existed became the stage on which Earth's most consequential transformation began, quietly, invisibly, without witnesses. At some indeterminate moment, chemistry crossed a threshold from which it would never return. The only question is where that boundary truly lies, and whether it was ever a boundary at all. For centuries, life was defined by what it looked like: movement, growth, breath. But science eventually stripped those impressions away and asked a harder question: what does life do? At its most precise, life is not a substance or a shape. It is a process, a system capable of maintaining itself, drawing energy from its surroundings, and, crucially, producing imperfect copies of itself over time. Without replication, there is no evolution. Without evolution, complexity has no mechanism to accumulate. This distinction matters because it allows us to trace life back before cells, before membranes, even before biology itself. Long before the first organism existed, Earth's oceans were filled with organic molecules. Amino acids, nucleotides, lipids, the chemical building blocks of life, form naturally under a wide range of conditions. Laboratory experiments and meteorite analyses confirm this repeatedly. These molecules are not rare. They are a predictable outcome of carbon chemistry given energy and time. But building blocks alone do not make life. The central problem of abiogenesis is not creation. It is organization. Organic molecules must be arranged into systems that persist. They must resist dilution, degradation, and randomness. Most chemical reactions move toward equilibrium, toward sameness. Life does the opposite. It maintains difference. The leading framework for explaining how this began is known as the RNA world hypothesis. It is not a claim that RNA was alive, but that it occupied a critical transitional role. RNA is unusual. Unlike DNA, which stores information, and proteins, which perform chemical work, RNA can do both. Certain RNA molecules, ribozymes, can catalyze reactions, including the assembly of other RNA strands. In controlled experiments, RNA has been shown to replicate segments of itself, imperfectly but persistently. This dual role solves a fundamental paradox. Modern life separates information storage from metabolism. Early systems could not afford that division. RNA offers a plausible bridge, a molecule capable of carrying instructions while actively participating in chemistry. Still, RNA does not arise spontaneously as a complete system. It requires concentration, stability, and energy. This is where environment becomes decisive. Mineral surfaces likely played a key role. Clays and metal sulfides can bind organic molecules, holding them in proximity long enough for reactions to occur. On open water, molecules diffuse and disperse. On surfaces, they accumulate. Experiments show that certain minerals promote the formation of longer, more complex chains of nucleotides. Energy gradients provided the drive. At hydrothermal vents, chemical disequilibria persist naturally. Hydrogen-rich fluids meeting carbon dioxide-rich seawater, generating reactions without external intervention. These environments supply continuous energy, not in bursts, but as steady flows. Life requires this constancy. Membranes added another layer of control. Simple lipid molecules spontaneously form vesicles in water, enclosing small volumes. These structures are not alive, but they create inside and outside, a prerequisite for regulation. When reactive molecules become trapped inside such compartments, their interactions become more frequent, more selective. At some point, systems emerged that could do something unprecedented. They could make copies, not perfect copies. Perfect replication would freeze change. Instead, early replication was sloppy. Errors occurred. Variants formed. Most failed. A few persisted longer than others. This is where replication overtakes metabolism in importance. Metabolism allows a system to function. Replication allows it to continue. A chemically active system that cannot reproduce vanishes when conditions change. A replicating system, even a fragile one, can survive catastrophes by producing many imperfect descendants. Once replication exists, selection is unavoidable. No intention is required, no direction. Variants that persist longer or reproduce more efficiently become more common, those that do not disappear. Over time, complexity accumulates, not because it is favored, but because it sometimes works. It is important to be precise here. There is no single moment when chemistry became life. There is no universally agreed boundary. Instead, there is a gradient, systems becoming increasingly lifelike as replication, compartmentalization, and energy use become integrated. Science does not yet know which exact pathway led to the first true ancestor of all life. Competing models exist: metabolism-first scenarios, lipid-first systems, hybrid networks combining multiple processes. Each is supported by partial evidence. None require miracles, none violate physical law. What they share is this conclusion: life emerged not through a single leap, but through accumulation. Replication changed everything. Because once matter can copy itself, the future is no longer a repetition of the past. It becomes a branching tree of possibilities. Survival becomes conditional. Resources become contested. Stability becomes temporary. And from that moment onward, the planet is no longer governed by chemistry alone. If you're finding this journey valuable, if you appreciate an exploration of life grounded in evidence rather than spectacle, consider subscribing. This story unfolds slowly, deliberately, and every step builds toward what comes next. Because replication does not merely allow survival, it creates competition. And competition will soon begin reshaping life in ways that even chemistry could never predict. The earliest life on Earth did not announce itself. It left no bones, no shells, no obvious shapes. What it left instead were chemical fingerprints, subtle, persistent, and extraordinarily old. By at least 3.7 to 3.8 billion years ago, the planet was no longer lifeless. This conclusion does not come from speculation, but from converging lines of geological evidence. In ancient rocks from Greenland, Australia, and South Africa, scientists have identified carbon isotopic ratios that strongly favor biological activity. Living systems preferentially use lighter carbon isotopes. When those ratios appear locked inside rocks billions of years old, they point to metabolism, not chemistry alone. In some locations, the evidence is even more tangible. Layered structures known as stromatolites rise from ancient sedimentary deposits, fossilized microbial communities that once lived in shallow waters. These formations are not accidental. They are created when microbial mats trap sediments, grow upward toward light, and repeat the process over countless generations. Modern versions still exist today. Their ancient counterparts, preserved in stone, are among the oldest macroscopic records of life on Earth. These organisms were simple, but they were not fragile. The first life forms were single-celled microbes, lacking nuclei, organelles, or complex internal structures. They were prokaryotes, bacteria and archaea, and they operated at the smallest viable scale. Their genetic material floated freely within the cell. Their metabolisms were efficient, flexible, and resilient. They did not rely on oxygen. In fact, oxygen was toxic to them. Instead, early microbes drew energy from chemical reactions involving hydrogen, sulfur, iron, and carbon compounds. Some harvested energy from sunlight without producing oxygen. Others thrived in darkness deep beneath the ocean floor, fueled entirely by geochemistry. Their diversity was metabolic, not anatomical. This matters because it explains why life survived. Early Earth was still unstable. Impacts occurred, temperatures fluctuated, oceans shifted. But microbial life was adaptable. When conditions worsened in one environment, microbes persisted in another. Heat, pressure, acidity, radiation. None of these were absolute barriers. They were variables. At this stage, life was not advancing toward complexity. It was consolidating its presence. Genetic evidence allows scientists to infer the existence of a common ancestor from which all modern life descends. This organism is known as LUCA, the last universal common ancestor. LUCA was not the first living thing. It was the most recent organism whose descendants include every bacterium, archaeon, plant, animal, and human alive today. Importantly, LUCA was not a single, isolated cell living alone in a pond. It was part of a population, a network of organisms exchanging genes, competing for resources, and evolving together. Early life did not evolve as a clean, branching tree. It evolved as a tangled web. From genetic reconstruction, Luca appears to have been surprisingly sophisticated. It likely used DNA to store information, RNA to translate it, and proteins to perform chemistry. It possessed enzymes, membranes, and metabolic pathways that remain fundamental to life today. It likely lived in an environment rich in hydrogen and metals, possibly near hydrothermal systems. What Luca was not is equally important. It was not complex in structure. It was not multicellular. It did not think, sense, or plan. It did not represent a pinnacle. It represented a baseline, a minimal system that worked well enough to persist. Evolution at this stage was constrained by physics and chemistry. Energy availability limited size. Diffusion limited complexity. Genetic innovation occurred, but slowly. Horizontal gene transfer, the sharing of genetic material between unrelated organisms, was common, blurring distinctions between lineages. Survival favored efficiency, not elaboration. And yet, despite these constraints, life spread. Microbial communities colonized oceans, sediments, and subsurface environments across the planet. They formed biofilms, mats, and layered ecosystems. They altered local chemistry, changing pH, mineral composition, and gas concentrations. In doing so, they began interacting with the planet itself. This is a critical shift. Life was no longer merely adapting to Earth. It was modifying it. Microbes altered the cycling of carbon, sulfur, and nitrogen. They influenced rock formation. They mediated chemical reactions that would not occur spontaneously. Over time, their cumulative activity began to register at planetary scale. But this influence was slow, almost imperceptibly so. For hundreds of millions of years, life remained microscopic. There were no predators in the familiar sense, no eyes, no movement beyond diffusion and simple motility. Evolution proceeded, but cautiously, constrained by energy, environment, and competition among equally simple organisms. This was not a failure of innovation. It was a period of stabilization. Life had crossed the threshold from chemistry into biology. Now it needed to endure, to explore metabolic possibilities, to establish a foothold strong enough to survive planetary change. And it succeeded. By the time life reached its first billionth year, it was no longer rare or localized. It was global, persistent, embedded into the functioning of Earth itself. If this exploration of life's earliest foundations is something you value, if you prefer clarity over exaggeration and evidence over mythology, consider subscribing. The story unfolds step by step, and each step builds directly on what came before. Because these early organisms, invisible and unremarkable by modern standards, were not merely surviving. They were preparing the planet for a transformation so profound that it would nearly destroy them. And in doing so, they would reshape Earth forever. For most of Earth's history, life had no faces. No forests, no animals, no movement visible to the naked eye. For more than 2 billion years, nearly half the age of the planet, Earth was a microbial world. Bacteria and archaea did not merely exist during this time, they dominated every environment that could sustain chemistry. This long interval is sometimes mischaracterized as evolutionary stagnation. In reality, it was one of the most innovative periods in the history of life. Microbes explored nearly every viable way to extract energy from their surroundings. Without oxygen, without sunlight in many cases, and without complex structures, they diversified metabolically rather than anatomically. Some organisms fermented organic molecules, breaking them down to release small amounts of energy. Others evolved methanogenesis, a metabolism found only in archaea, producing methane by combining hydrogen and carbon dioxide. This process altered the atmosphere, contributing to greenhouse warming and helping keep the planet from freezing under a fainter young sun. Still others mastered an oxygenic photosynthesis, capturing light energy without producing oxygen. These microbes use compounds like hydrogen sulfide or iron instead of water, creating localized chemical gradients in oceans and sediments. Entire ecosystems functioned around these metabolisms, unseen but extensive. This biochemical creativity explains why microbes endured. Rather than waiting for environments to stabilize, they adapted to instability itself. When one energy source became scarce, another pathway emerged. When conditions shifted, metabolism shifted with them. Genetic innovation did not occur only through slow mutation. Early life relied heavily on horizontal gene transfer, the direct exchange of genetic material between organisms, even across species boundaries. Genes moved laterally through populations, spreading successful solutions rapidly. This process fundamentally altered evolution's tempo. Instead of waiting generations for advantageous traits to spread through reproduction, microbes shared them almost instantly. Metabolic pathways, stress responses, and resistance mechanisms propagated across ecosystems. Early evolution was communal before it became competitive. The result was a biosphere that functioned more like a network than a tree. This genetic fluidity also explains why defining early species is difficult. Boundaries were porous, lineages merged, separated, and merged again. What mattered was not ancestry alone, but functionality. Microbial life also demonstrated an extraordinary tolerance for extremes. Some thrived at temperatures exceeding boiling water. Their proteins stabilized against heat that would destroy modern cells. Others endured highly acidic environments, alkaline lakes, or intense radiation. Many survived deep beneath the surface, isolated from sunlight entirely, sustained by chemical reactions within rock. These extremophiles are not evolutionary curiosities. They represent the rule, not the exception, for early life. Earth's surface was still unstable. Oceans fluctuated in chemistry. Impacts and volcanism reshaped environments repeatedly. Life that survived had to be resilient first and efficient second. This resilience came at a cost. Complexity requires energy. It requires stable conditions, predictable resources, and surplus metabolic capacity. Microbial life optimized for survival in extremes did not benefit from becoming larger, slower, or more structurally elaborate. Simplicity was not a limitation, it was an advantage. This is why complexity took so long to appear. For billions of years, microbes had no incentive to build bodies. Single cells with adaptable metabolisms were better suited to the planet as it was. Evolution does not aim toward complexity. It responds to conditions, and for most of Earth's history, complexity was unnecessary. But innovation did not stop. Within this microbial world, one lineage developed a metabolism that would change the planet irreversibly. A method of capturing sunlight that split water molecules, releasing oxygen as waste. At first, this oxygen reacted immediately with surrounding minerals and dissolved iron. It did not accumulate, but production continued. Gradually, sinks filled, reactions saturated, oxygen began to persist. What had once been a rare and reactive byproduct became an environmental force. For the microbes that produced it, oxygen was incidental. For most life on Earth, it was lethal. This single metabolic invention would trigger a planetary transformation, one that extinguished countless lineages, altered atmospheric chemistry, and rewrote the rules of survival. If you're finding value in understanding life through evidence rather than assumption, and complexity through process rather than purpose, consider subscribing. Each step in this story builds directly from the last, revealing how small biochemical changes can reshape entire worlds. For nearly 2 billion years, Earth's atmosphere had been a silent, oxygen-free world. Life thrived beneath its thin, hazy skies, not through photosynthesis that produced oxygen, but through metabolisms that relied on chemical compounds such as hydrogen, sulfur, and methane. In these early oceans, cyanobacteria had already evolved the ability to harness sunlight, but at first, their energy capture was limited. Then, somewhere between 2.7 and 2.5 billion years ago, a lineage of cyanobacteria perfected a process that would irrevocably change the planet: oxygenic photosynthesis. This metabolism was radical in its simplicity. Using water, carbon dioxide, and sunlight, cyanobacteria split water molecules, releasing oxygen as a byproduct. At first, this oxygen was immediately consumed. Dissolved iron in seawater bound the free oxygen, forming massive deposits of iron oxide that would later crystallize into banded iron formations, one of the most conspicuous geological signatures of early life. These formations, preserved in ancient rock, record the first evidence that life could alter the chemistry of the entire planet. For millions of years, oxygen production continued, but the planet absorbed it quietly. Eventually, the reactive sinks, iron, sulfur, and other reduced compounds, were filled. Oxygen could no longer be removed as quickly as it was produced. The atmospheric concentration of oxygen began to rise. This gradual accumulation, occurring around 2.4 billion years ago, is what geologists call the Great Oxidation Event. The consequences for life were immediate and severe. For organisms adapted to oxygen-free environments, oxygen was highly toxic. Its reactive chemistry damages proteins, lipids, and nucleic acids. Anaerobic microbes, which had dominated the biosphere for billions of years, faced extinction on a planetary scale. Estimates suggest that this was the largest mass extinction in Earth's early history, removing perhaps 90% of existing microbial species. Yet oxygen also enabled innovation. Its presence allowed the development of new biochemical pathways. Aerobic respiration emerged, a metabolism vastly more efficient than fermentation or other anaerobic processes. Organisms could now extract far more energy from the same amount of food, enabling larger cells and eventually more complex multicellular forms. Oxygen was both a poison and an opportunity, acting as a selective pressure that reshaped ecosystems and opened the door to complexity. Geochemical evidence preserves the details of this transformation. The iron deposited in banded formations ceased once oceans became saturated with oxygen, leaving a distinct record in sedimentary rock. Sulfur isotopes indicate fluctuations in ocean chemistry, reflecting shifts in microbial populations and oxygen levels. Carbon isotopes show evidence of widespread changes in photosynthetic activity, marking a planet in transition. These records, spanning multiple continents and preserved in rocks billions of years old, provide an extraordinary testament to the scale and impact of the event. The rise of oxygen also had profound atmospheric consequences. Oxygen reacts readily with ultraviolet radiation, forming ozone. By creating an ozone layer in the upper atmosphere, oxygen began to shield the surface from harmful ultraviolet light, a protective mechanism that would later enable life to venture onto land. But the ozone layer was not immediate. It developed gradually as oxygen concentrations reached sufficient levels. Until then, surface environments remained hazardous to newly emerging organisms, sensitive to radiation. This period also illustrates the fundamental principle that extinction drives opportunity. The organisms that survived the oxygenation event were those capable of using oxygen or retreating into niches where oxygen remained scarce. They carried the molecular innovations necessary to exploit this new chemistry. Over time, these survivors diversified, setting the stage for subsequent evolutionary leaps. The rise of oxygen reshaped the biosphere from microbial simplicity to ecological complexity, but it did so by destroying nearly all that had come before. Oxygenic photosynthesis also transformed the geochemistry of the oceans. Dissolved iron decreased dramatically as oxidation continued, changing the chemical gradients available to life. This affected the distribution of nutrients, the formation of minerals, and the overall cycling of elements critical to biology. Ocean chemistry was no longer static. It was dynamic, influenced directly by biological activity. Life was no longer a passive passenger on Earth. It had become a planetary engineer. The Great Oxidation Event represents the first time life created a biosphere-wide change so profound that the planet itself was remade. The scale of this transformation is difficult to grasp. Microbes, invisible to the eye, produced enough oxygen over millions of years to fundamentally alter the chemistry of oceans and atmosphere, eliminate dominant species, and establish conditions for larger, more complex organisms. It is one of the earliest examples in Earth's history where biology and geology are inseparably intertwined. Yet oxygen came with limitations. Aerobic metabolism allowed energy-intensive cellular processes, but it also introduced oxidative stress, forcing organisms to develop protective mechanisms, repair enzymes, and sophisticated regulatory systems. These adaptations were prerequisites for later complexity, but also imposed new constraints. Life was becoming more capable, but it was also more delicate. During this time, the surface of the planet remained largely microbial. The first eukaryotic cells, which would later give rise to complex life, appeared only after oxygen concentrations had stabilized, suggesting that the Great Oxidation event created both the challenge and the opportunity necessary for cellular innovation. Without the oxygenation of the planet, eukaryotes, multicellular life, and eventually animals and humans might never have emerged. The Great Oxidation event also illustrates a recurring theme in Earth's history. Planetary transformations often emerge from small biological innovations acting over vast timescales. A single metabolic process, splitting water to extract electrons, led to an extinction, a reconfiguration of oceans and atmosphere, and the eventual rise of complex life. It demonstrates that biology is both reactive and creative, a force that adapts to circumstances while simultaneously shaping those circumstances on a planetary scale. By 2.0 billion years ago, oxygen levels had stabilized at low but persistent concentrations. The biosphere had been reshaped. Anaerobic ecosystems survived only in isolated or protected niches, while oxygen-using organisms diversified. Life's chemistry had fundamentally changed. It was now possible for cells to grow larger, store energy more efficiently, and develop internal compartments, paving the way for eukaryotes and, ultimately, multicellular life. The story of oxygen is not merely one of survival or extinction. It is a story of planetary coevolution, in which life and environment are inseparable. The atmosphere became a reflection of biology itself, and the rules of survival were rewritten. What had once been a simple microbial world was now poised for a scale of complexity previously unimaginable. Oxygen made larger life possible, but with size came new challenges, energy demands, vulnerability to damage, and the need for coordination among cells. These constraints would shape the evolution of eukaryotes, multicellular organisms, and eventually the first animals. Earth had learned a fundamental lesson. Innovation carries both destruction and opportunity, and the consequences of a single microbial invention can resonate across billions of years. This planetary oxygenation set the stage for the next phase of life, where cells would cooperate, specialize, and explore new forms. A transformation from survival as a chemical process to survival as an organized, dynamic system. For nearly 2 billion years after the rise of oxygen, life remained microbial. Bacteria and archaea dominated oceans, sediments, and extreme environments, their diversity metabolic rather than structural. They had survived extinction, reshaped the planet, and experimented with chemical innovation, yet the vast majority remained microscopic and simple. It was efficient, resilient life, but it was constrained. Energy, not intelligence, had been the limiting factor. Cells were too small, too minimalist, and too uniform to build anything larger. Then, somewhere around 1.8 to 1.5 billion years ago, a radical transformation occurred, the emergence of the eukaryotic cell. Unlike its prokaryotic predecessors, the eukaryotic cell was internally divided, compartmentalized into specialized organelles, each performing dedicated functions. It had a nucleus to protect genetic information, endomembrane systems to shuttle proteins and lipids, and cytoskeletal elements that could support shape and movement. This was not just a step in evolution, it was a structural revolution, one that allowed life to escape the energetic constraints of prokaryotic simplicity. At the heart of this transformation lies the theory of endosymbiosis. Eukaryotic cells, according to this theory, arose when certain prokaryotic cells entered into a symbiotic relationship within another cell. Instead of being digested or destroyed, these internalized cells persisted, forming a mutually beneficial arrangement. The most famous examples are mitochondria and later chloroplasts. Mitochondria, derived from an ancestral alpha proteobacterium, became cellular powerhouses, producing energy in the form of ATP through aerobic respiration. Chloroplasts, descending from cyanobacteria, enabled photosynthesis in plant cells, allowing sunlight to be captured directly to fuel metabolism. The acquisition of mitochondria represented more than just a new organelle. It was a dramatic expansion of the cell's energy budget. Prokaryotic cells are limited in size because energy production is constrained by the surface area of the plasma membrane. By internalizing energy-generating symbionts, early eukaryotes could support larger volumes, more complex structures, and increased genomic content. More energy meant more possibilities. Cells could maintain internal compartments, regulate reactions precisely, and store or process information on a scale previously impossible. With increased energy came genomic complexity. Eukaryotic genomes expanded, not just in size, but in regulation. Genes could be turned on and off in sophisticated patterns. Introns and exons allowed alternative splicing, producing multiple proteins from a single gene. Regulatory sequences controlled timing, location, and intensity of expression. Internal membranes allowed compartmentalization of biochemistry, enabling incompatible reactions to occur simultaneously without interference. This modularity created flexibility, an ability to respond to changing environments in ways that simple prokaryotic cells could not. Yet eukaryotes did not emerge fully formed. Early eukaryotes were likely slow-growing, vulnerable, and small relative to modern cells. Their innovation was in principle, not perfection. The process required not intelligence, but the gradual alignment of metabolism, energy availability, and molecular compatibility. Endosymbiosis is neither miraculous nor instantaneous. It is a product of persistence and opportunity, of chemistry and competition acting over millions of years. Importantly, eukaryotic evolution illustrates a central lesson of life. Complexity is always a matter of energy. Prokaryotic life could survive for billions of years without building large structures. It did not lack ingenuity. It lacked the energetic capacity to support cellular elaboration. Once that barrier was overcome, once energy became abundant and efficiently usable, life could afford to expand, compartmentalize, and diversify. Innovation follows energy, not intention. The rise of eukaryotes also introduced cooperation at the cellular level. Endosymbiotic relationships required negotiation, one cell living inside another, sharing resources and avoiding conflict. These early cooperative interactions foreshadowed later multicellularity. They were experiments in coordination, in building systems where independent units work together for mutual benefit. Cooperation at this scale would eventually allow bodies to form rather than solitary cells. Eukaryotic cells also set the stage for specialization. Organelles enabled partitioning of functions. Energy production, protein synthesis, waste processing, and storage could all occur in parallel. By isolating incompatible chemical reactions, cells could perform multiple complex processes simultaneously. This created the potential for differentiation, one of the prerequisites for multicellular life. Cells could now evolve into specialized roles rather than remaining generalist. The emergence of eukaryotes also had ecological consequences. They introduced new metabolic pathways, exploited oxygen-rich environments more effectively, and competed in ways prokaryotes could not match. Their presence increased the overall efficiency of ecosystems, reshaping nutrient cycles and altering the chemical balance of oceans. At a microscopic scale, they were agents of innovation, and at a planetary scale, they subtly rewrote the rules of life. Eukaryotic evolution was neither fast nor linear. Fossil evidence suggests that these cells were rare for hundreds of millions of years before they diversified. Molecular clocks indicate that the first true eukaryotic lineages arose perhaps a billion years before the first animals. Complexity required patience, energy, and stability. It was a slow experiment in building systems capable of more than survival, systems capable of cooperation, growth, and information processing. By the end of this interval, the biosphere was still overwhelmingly microbial, yet eukaryotic cells, with their internal compartments, mitochondria, and regulatory sophistication, represented a turning point. Life had acquired the structural capacity to do more than endure. It could store energy, manage resources, and coordinate internally on a scale that made larger, multicellular organisms feasible. This was the true beginning of advanced life, not in animals, not in forests, not even in visible complexity, but in cells that could harness energy, maintain internal order, and cooperate with others to achieve something greater than themselves. These innovations laid the groundwork for bodies, for tissues, for organs, the architecture of all subsequent multicellular life. The story of eukaryotic cells demonstrates a critical principle. Evolution does not favor intelligence or foresight. It favors energy and persistence. The cell that changed everything was not clever, it was capable, it was organized, it was prepared. Once cells learned to cooperate, to share energy and information internally and externally, the stage was set for a transformation that would surpass anything microbes could achieve alone. Life was no longer constrained by minimalism. The potential for form, function, and size had arrived. With the advent of eukaryotic cells, life had acquired the capacity for internal complexity, energy-intensive processes, and cooperation. Yet these innovations alone did not immediately produce large structures. For millions of years, eukaryotes existed primarily as single cells, capable of differentiation only internally or in temporary colonies. The next major transition was the evolution of multicellularity, the aggregation of individual cells into persistent, coordinated bodies. Multicellularity did not arise once and never again. It evolved independently in multiple lineages, including animals, fungi, plants, and various algal groups. The repeated emergence of this trait highlights a key principle. When energy availability, cellular complexity, and environmental conditions converge, natural selection repeatedly discovers similar solutions. In essence, multicellularity is a probable outcome under certain biochemical and ecological circumstances, not a singular miracle. At its simplest, multicellularity required three components: cells adhering to one another, cells communicating to coordinate behavior, and the ability to differentiate into distinct types. Adhesion could be mediated by extracellular proteins or polysaccharides, ensuring that cells remained attached after division. Communication relied on chemical signals, molecules that allowed cells to sense neighbors, transmit instructions, and respond collectively. Differentiation allowed subsets of cells to specialize for locomotion, reproduction, or nutrient acquisition, increasing overall efficiency. The earliest multicellular organisms were modest. Fossil evidence from around 1.6 billion years ago suggests the presence of filamentous algae and simple colonial structures. Cells adhered, divided labor, and persisted as collective units, yet they remained microscopic and largely uniform. These proto-organisms demonstrate that multicellularity began not as a leap in size, but as a leap in cooperation. The transition to more complex multicellularity required innovation at the genetic level. Regulatory genes evolved that could orchestrate development, determining when cells should divide, die, or differentiate. Some of the earliest developmental genes are recognizable today across diverse multicellular lineages. Their presence indicates that the blueprint for differentiation existed long before animals or plants appeared. Life was learning not only to survive but to organize itself spatially and temporally. Cellular specialization, however, introduced new constraints. Differentiated cells often lost autonomy, becoming dependent on the collective. Reproductive cells were distinct from somatic cells, immune and digestive functions required division of labor, and structural cells consumed resources without contributing directly to reproduction. Multicellularity was a compromise. Specialization increased efficiency and size, but increased vulnerability to failure. Damage to one part of the organism could threaten the whole. Life had gained power, but with it, fragility. The rise of early animals and algae illustrates these dynamics. Fossils from the Mesoproterozoic and Neoproterozoic eras show evidence of organized cell layers, repeated structures, and coordinated growth patterns. These organisms were still small, but they exhibited features essential for future complexity: differentiation, polarity, and pattern development. They demonstrate that even at a microscopic scale, evolution experiments with form as a functional solution. Environmental pressures likely reinforced the transition. Larger multicellular bodies could exploit new niches, capturing light, filtering water, or moving more effectively in search of nutrients. Cooperative cells could resist predation or tolerate fluctuating conditions better than single cells. Ecological interactions created selective advantages for groups that could coordinate and specialize. Life was learning not only to survive individually but to exploit collective strategies. At the molecular level, multicellularity required innovations in adhesion, signaling, and extracellular matrix production. Proteins such as cadarins and integrins facilitated cell attachment and communication, while regulatory networks controlled timing and differentiation. These systems were not static, they evolved incrementally, shaped by mutation, selection, and horizontal gene transfer. Each successful adaptation allowed multicellular assemblies to grow larger, last longer, and respond more effectively to environmental cues. The evolutionary path to true multicellular animals remained long and constrained. Most lineages experimented with simple forms, sheets of cells, filaments, hollow spheres, before more complex body plans emerged. Some lineages reverted to unicellularity, demonstrating that multicellularity was not universally advantageous. Success depended on energetic efficiency, structural integrity, and the ability to reproduce reliably. Evolution tested these factors repeatedly, producing a diverse array of solutions before animals became dominant. This period also saw the refinement of developmental control. Regulatory genes allowed repeated structures, segmented bodies, and polarity along axes. Morphogens, chemical signals that guide development, emerged, establishing gradients that dictated cell fate. This genetic and biochemical scaffolding allowed life to move from simple colonies to coordinated organisms with predictable architecture. By the end of the Proterozoic, multicellular life had achieved a degree of organization unprecedented in Earth's history. Cells were no longer solitary units, they were components of bodies, capable of division of labor, specialization, and cooperation over extended periods. Ecological interactions intensified. Microbes were no longer the only architects of chemical and geochemical change. Multicellular organisms could manipulate their environments more directly, laying the groundwork for food webs, predation, and niche construction. Multicellularity illustrates a fundamental evolutionary principle. Complexity requires coordination and energy, not foresight. Bodies are compromises, structures optimized to balance survival, reproduction, and efficiency. They are powerful, yet fragile. They allow organisms to exploit new environments and acquire new functions, but they also introduce vulnerabilities absent in single-celled life. Evolution does not engineer perfection. It assembles workable solutions that persist. By combining cooperation, differentiation, and regulation, life achieved a capability no single cell could possess. Bodies could now grow, move, and respond collectively, providing the mechanical and ecological foundation for later innovations. The first multicellular organisms were small and simple, but they transformed the potential of life. For the first time in Earth's history, life became visible. Its presence was no longer confined to microscopic mats, sediments, or chemical signatures. Bodies existed, modest, fragile, and experimental, but bodies that could interact with the world in new ways, manipulating light, fluid, and nutrients. From these early experiments, evolution would eventually construct the intricate tapestry of plants, animals, and ecosystems that define the modern biosphere. By the late Neoproterozoic, multicellular life had already experimented with form and function for hundreds of millions of years. Colonial algae and simple organisms had established the principles of adhesion, differentiation, and regulation. Yet visible animals remained rare, enigmatic, and largely immobile. The fossil record from this period, the Ediacaran biota, roughly 635 to 541 million years ago, shows soft-bodied forms, often flattened, lacking obvious organs or appendages. These were experiments in size and symmetry, some with fractal-like branching patterns, others with segmented or radial designs. They left impressions in sedimentary rock but little evidence of movement, predation, or complex interactions. The Ediacaran world was dominated by sessile or slow-moving organisms. Most absorbed nutrients directly from the water, either by diffusion or surface feeding. Competition existed, but it was largely local, confined to patchy microbial mats and nutrient gradients. Energy budgets were limited and mobility was constrained. There were no skeletons, no eyes, no specialized appendages, just bodies exploring the possibilities of multicellularity in a chemically and physically hospitable ocean. Then, at the dawn of the Cambrian period, around 541 million years ago, the pace of evolution accelerated dramatically. What paleontologists call the Cambrian explosion was not instantaneous creation, but a rapid diversification over tens of millions of years. During this interval, nearly all major animal body plans recognizable today emerged. Segmentation, appendages, and coeloms became common. Organ systems, including primitive nervous and circulatory structures, began to appear. Key to this diversification was energy. By this time, oxygen levels in the oceans had stabilized, providing sufficient metabolic power to support larger, more active bodies. Mitochondria-enabled energy budgets allowed organisms to invest in movement, sensing, and predation, functions that had previously been prohibitively costly. Predation itself created selective pressures, favoring defensive structures, speed, and the ability to detect and respond to threats. This feedback loop, attack and defense, accelerated evolutionary innovation, creating an ecological arms race that reshaped marine ecosystems. Skeletons emerged as both armor and support. Calcium carbonate and chitin allowed bodies to grow larger, resist mechanical stress, and exploit new ecological niches. Hard parts preserved in the fossil record make the Cambrian particularly visible to us today, providing a window into the rapid rise of animal diversity. Eyes appeared independently in multiple lineages, facilitating navigation, hunting, and predator avoidance. Even simple, light-sensitive structures conferred significant advantage, intensifying selection for sensory sophistication. Mobility transformed the oceans. Organisms no longer drifted passively with currents. They swam, burrowed, or crawled, creating new ecological opportunities and pressures. Sediment mixing, nutrient cycling, and predation rates increased. The biosphere became an interconnected system, in which actions of one organism could influence many others. These interactions promoted specialization, coevolution, and further complexity. Despite this rapid diversification, body plans quickly stabilized. Natural selection favored structural solutions that were effective, efficient, and repeatable. Once a functional arrangement of tissues and organs proved successful, it persisted and diversified rather than being continuously replaced. This explains why the major animal phyla have endured for hundreds of millions of years with relatively little fundamental change. Evolution optimized the toolbox. It did not reinvent it with each generation. The Cambrian explosion also highlights the cumulative nature of evolutionary innovation. Oxygen, eukaryotic complexity, and multicellular cooperation set the stage. Each advance provided the energetic and structural foundation for the next. The evolution of predation, skeletons, and sensory organs was only possible because cells could coordinate, metabolize efficiently, and maintain larger bodies. Complexity emerged stepwise, each stage constrained and enabled by preceding innovations. Life in the Cambrian was both spectacular and fragile. While diversification was rapid, extinction rates were high, and ecological competition intense. Many lineages disappeared, leaving only those that successfully balanced energy acquisition, reproduction, and environmental adaptation. The survivors carried forward the innovations that would underpin future terrestrial and aquatic ecosystems. By the end of the Cambrian, oceans were no longer simply filled with microbes and simple multicellular forms. They hosted complex food webs, interactive communities, and active predation. Life had learned to move, to sense, to exploit resources, and to compete on a scale never before seen. Evolution had accelerated, producing a diversity of forms and behaviors that would persist and expand for hundreds of millions of years. The Cambrian marks the first time life becomes fully visible in the fossil record as more than chemical traces or impressions. It demonstrates that evolutionary innovation can proceed rapidly once energy, oxygen, and structural capacity align. It shows the power of selection to accelerate complexity when ecological interactions intensify. And it establishes the pattern that would define animal evolution: bodies as tools, energy as currency, and natural selection as the unrelenting sculptor. The next stage of life's history would require leaving the oceans entirely. Life would confront gravity, desiccation, and ultraviolet radiation in ways previously irrelevant. The first experiments on land would challenge the principles that had governed oceans for billions of years, creating new pressures and opportunities that would redefine the trajectory of evolution. The oceans had nurtured life for over 3 billion years. Within them, oxygen stabilized, eukaryotes multiplied, multicellularity emerged, and complex animals diversified. Yet Earth's surface remained largely uninhabited. Rocks, deserts, and shallow shorelines were lifeless. The transition from water to land was not inevitable. It was a formidable challenge, demanding solutions to a host of new physical, chemical, and ecological problems. Gravity, desiccation, and ultraviolet radiation transformed the environment from a supportive medium into a relentless obstacle course. The first organisms to experiment with terrestrial life were plants and fungi, acting not independently, but as cooperative pioneers. Early non-vascular plants, such as moss-like bryophytes, colonized moist surfaces. Their success depended on symbiotic relationships with fungi, mycorrhizal networks that penetrated soils, scavenging minerals and water while providing carbohydrates in return. This partnership allowed plants to survive in nutrient-poor substrates and laid the foundation for soil formation. By breaking down rock, trapping organic matter, and cycling nutrients, these early colonists fundamentally altered Earth's surface. Land became chemically habitable. The colonization of land was accompanied by atmospheric feedback. Plants released oxygen through photosynthesis, increasing local and eventually global O2 concentrations. Soil development facilitated carbon sequestration, altering the balance of greenhouse gases and stabilizing climate over geological timescales. Life was not merely occupying land, it was engineering it, turning barren surfaces into productive ecosystems capable of sustaining increasingly complex organisms. Fungi, often invisible, played an equally critical role. They decomposed organic matter, stabilized soil, and mediated nutrient cycles. Without them, plants could not have thrived, and soils would have remained thin and infertile. Together, plants and fungi established the structural and chemical foundation for life to expand beyond the water. The first animals to venture onto land were arthropods, small exoskeleton-bearing organisms that retained moisture and could resist desiccation. Insects and myriapods experimented with locomotion, respiratory adaptations, and sensory integration. Their exoskeletons provided structural support against gravity, while internal tracheal systems allowed efficient gas exchange without losing water. These innovations enabled movement and feeding in an environment far less forgiving than the oceans. Tetrapods emerged later, representing a more radical transition. Descending from lobe-finned fish, early tetrapods adapted fins into limbs capable of supporting weight on solid ground. Lungs evolved to extract oxygen from air, and skin developed to limit water loss. Early amphibians remained tied to moist environments, often reproducing in water, but they demonstrated the feasibility of terrestrial locomotion and exploitation of new habitats. Gravity imposed limits on body size, locomotion, and structural organization, forcing evolution to reinvent skeletal and muscular systems. The colonization of land highlights the interplay between opportunity and constraint. Terrestrial environments offered abundant resources: light, oxygen, and unoccupied space. Yet they were lethal without adaptation. UV radiation damaged DNA, fluctuating temperatures stressed metabolism, and desiccation threatened cellular integrity. Success required innovations across multiple levels: structural, biochemical, and behavioral. Early terrestrial ecosystems were fragile and localized. Plant mats, soil communities, and small arthropod populations formed mosaics of habitation rather than continuous biomes. Life had to innovate not only to survive, but to maintain continuity over space and time. Adaptation was not incremental improvement, it was reinvention, requiring the reorganization of physiology, morphology, and behavior. By altering land surfaces, early plants and fungi indirectly facilitated further animal colonization. Soil, stabilized and enriched by organic matter, provided surfaces for insects and tetrapods to feed, move, and reproduce. Oxygen-rich atmospheres enabled higher metabolic rates and larger body sizes. Life reshaped its own environment, creating feedback loops that reinforced terrestrial success. Structural challenges on land demanded continued innovation. Limbs evolved to support body weight against gravity, allowing coordinated locomotion. Vascular systems developed in plants to transport water from roots to leaves, while cuticles limited water loss. Respiratory adaptations, from tracheae to lungs, allowed efficient gas exchange in a medium far less dense than water. Vision and sensory organs adjusted to detect threats and resources in three-dimensional space above ground, rather than along a two-dimensional aquatic plane. By the late Paleozoic, terrestrial ecosystems had become remarkably diverse. Forests of early vascular plants interspersed with fungi created complex habitats. Arthropods dominated the soil, leaf litter, and canopy, while amphibians and early reptiles explored niches ranging from wetlands to river margins. The conquest of land was not instantaneous, but it represented a fundamental expansion of life's domain, a transformation in which energy, structure, and environmental modification converged to enable survival outside the oceans. This period demonstrates that terrestrial life is both powerful and perilous. Land offers opportunity, but survival is contingent on constant adaptation. Organisms cannot passively endure. They must actively engineer their physiology, morphology, and behavior to withstand extreme variability. Every step on land was a negotiation with gravity, radiation, and desiccation, a testament to life's capacity for innovation under constraint. Yet, even as terrestrial ecosystems expanded and stabilized, this dominance was precarious. Climatic shifts, geological upheavals, and fluctuating atmospheric conditions would repeatedly challenge these early colonists. Evolutionary success was not permanent. It required continual adjustment, a relentless process of experimentation and refinement that would shape the trajectory of life for hundreds of millions of years. Life had successfully crossed the boundary between water and land. From symbiotic partnerships between plants and fungi to the first tetrapod explorations, terrestrial habitats became inhabited, productive, and dynamic. Yet Earth's surface remained a battlefield, a place where survival demanded ingenuity, energy, and coordination. The conquest of land expanded the possibilities for life, but it also introduced fragility and dependence that would influence every organism to come. Life on Earth has never progressed in a steady, linear fashion. For every period of expansion and diversification, catastrophic setbacks have rewritten the rules, eliminating lineages and reshaping ecosystems. Mass extinctions are not anomalies. They are recurring features of the biosphere, dramatic punctuation marks in the history of life. These events demonstrate that destruction is not simply an end. It is a precondition for innovation. The fossil record identifies at least 5 major extinction events, the so-called Big Five, each reshaping life on a global scale. Among these, the Permian-Triassic extinction, approximately 252 million years ago, stands as the most devastating. Estimates suggest that up to 96% of marine species and 70% of terrestrial vertebrate species perished. Entire ecological systems collapsed, leaving vast voids in the biosphere. Life had been prolific, diverse, and geographically widespread, yet in geologically brief intervals, much of it disappeared. The causes of these extinctions are complex, often involving multiple interacting factors. In the case of the Permian-Triassic, immense volcanic eruptions in what is now Siberia released unprecedented quantities of carbon dioxide and sulfur dioxide into the atmosphere. The resulting greenhouse effect caused global temperatures to rise sharply, destabilizing climate patterns. Acid rain and ocean acidification further stressed ecosystems. At the same time, nutrient imbalances and ocean stagnation led to widespread anoxia, oxygen depletion, in the seas, suffocating organisms adapted to oxygen-rich environments. Extinction did not affect all life uniformly. Organisms with specialized physiologies, narrow habitats, or limited reproductive rates suffered most. Generalist species, small-bodied organisms, and those capable of surviving in extreme or fluctuating conditions were disproportionately represented among survivors. In this way, mass extinctions act as evolutionary filters, reshaping the gene pool and redirecting evolutionary trajectories. They remove previously dominant forms, creating ecological vacancies that favor previously minor or marginal lineages. The recovery from mass extinction is neither immediate nor uniform. Following the Permian to Triassic event, it took millions of years for ecosystems to stabilize. Recovery periods are characterized by evolutionary experimentation. New body plans, metabolic strategies, and ecological interactions emerge in response to emptied niches. Bottlenecks constrain genetic diversity, but they also focus selective pressure, accelerating the appearance of traits that confer adaptability. Evolution after extinction is opportunistic and rapid relative to the preceding equilibrium. Extinction events underscore a fundamental truth about evolution. It is not a process of steady improvement or accumulation of complexity. Success is contingent and ephemeral. Traits that confer dominance under one environmental regime may be fatal under another. What survives is not the strongest in absolute terms, but the most compatible with rapidly changing conditions. In this sense, catastrophe becomes a creative force, shaping life as powerfully as energy availability or cellular innovation. The Permian-Triassic extinction also altered the evolutionary landscape in specific, enduring ways. Many dominant Paleozoic marine invertebrates, such as trilobites and certain brachiopods, disappeared entirely. Their absence opened ecological space for new clades, including modern mollusks and crustaceans, to diversify. On land, the collapse of large amphibians allowed reptiles, particularly archosauriforms, to occupy ecological roles previously unavailable. These survivors would eventually give rise to the dominant vertebrate lineages of the Mesozoic, including dinosaurs. Mass extinctions demonstrate that ecological opportunity and innovation are tightly linked to prior catastrophe. Without the Permian-Triassic extinction, the evolutionary pathways leading to reptiles, archosaurs, and later mammals might have been blocked or delayed. The biosphere is continually reset by episodic destruction, and these resets are integral to the emergence of novel traits and body plans. Extinction, in other words, is not a failure of life, but a mechanism that enables evolutionary experimentation on a grand scale. The record of the Big Five also highlights the interplay between geological forces and biological response. Volcanism, tectonics, climate fluctuations, sea level changes, and atmospheric chemistry interact with evolutionary processes. Life is both resilient and constrained. Resilient in its ability to survive, adapt, and radiate after catastrophe. Constrained by physical limits, chemical boundaries, and ecological pressures. Mass extinction events reveal the biosphere as a dynamic system where destruction is inseparable from the potential for renewal. This principle repeats throughout Earth's history. Each time a lineage is removed, others are freed to diversify. Each ecosystem collapse is followed by a period of ecological experimentation, in which new strategies, forms, and interactions are tested against environmental realities. The survivors carry forward innovations, while previously dominant solutions are eliminated. Evolution, therefore, is not cumulative in a linear sense. It is punctuated, contingent, and profoundly shaped by loss. The Permian to Triassic extinction illustrates the extreme, yet even less catastrophic events demonstrate the same pattern. The late Devonian, Ordovician to Silurian, Triassic to Jurassic, and Cretaceous to Paleogene extinctions each reset ecological hierarchies, favoring new forms over old. Without these events, ecosystems might remain static, dominated by lineages that are incapable of further innovation. Extinction is, paradoxically, a prerequisite for evolutionary progress. Out of the devastation of the Permian to Triassic recovery, emerged organisms capable of exploiting the newly vacated niches. Small, versatile vertebrates expanded, eventually giving rise to the first archosaurs and eventually dinosaurs. Reptiles diversified in morphology and behavior, evolving locomotion, metabolism, and feeding strategies that would dominate terrestrial ecosystems for tens of millions of years. Life's capacity for adaptation, innovation, and expansion was intimately tied to the creative force of prior extinction. The history of mass extinctions teaches that life's trajectory is not linear. Catastrophe, far from halting evolution, acts as a mechanism for resetting ecological and evolutionary constraints. It accelerates the emergence of new traits, reshapes ecosystems, and redirects the flow of energy and diversity. In the aftermath of destruction, life experiments, innovates, and explores new solutions, often achieving forms, functions, and ecological dominance that would have been impossible under stable conditions. Out of the Permian collapse, life would rise again, more diverse and specialized than before. Reptiles would become giants, ecosystems more complex, and evolutionary potential expanded. Destruction had cleared the path for new forms, proving that extinction is not merely an end. It is an essential driver of innovation, shaping the trajectory of life across deep time. By the close of the Permian Triassic recovery, terrestrial ecosystems had been dramatically reshaped. The survivors were often small-bodied, adaptable, and capable of exploiting the open ecological spaces left by extinct lineages. Among these survivors were early archosauriforms, a group of reptiles that would set the stage for the Mesozoic, the age dominated by dinosaurs. Their evolutionary success was not a matter of inherent superiority, but of ecological opportunity, energy efficiency, and physiological flexibility. Archosaurs evolved in a world recovering from catastrophe, where competition was fragmented and resources were abundant relative to population density. Their bodies were built for endurance and adaptability. Limbs positioned under the body allowed efficient locomotion, enabling active predation and escape from threats. Respiratory systems were increasingly sophisticated, supporting higher metabolic rates. Their jaws and dentition diversified, facilitating omnivory, herbivory, and carnivory in ways earlier reptiles had not. These adaptations laid the foundation for the spectacular radiation of dinosaurs that would dominate the Triassic, Jurassic, and Cretaceous periods. Dinosaurs first appear in the fossil record approximately 230 million years ago in the late Triassic. The earliest forms were small, bipedal, and lightly built. They occupied niches that allowed them to move quickly and reproduce efficiently in a recovering landscape. Over the next tens of millions of years, they diversified rapidly, producing a remarkable array of body plans, sizes, and ecological strategies. Some became enormous quadrupeds capable of consuming vast amounts of vegetation, while others evolved into agile apex predators capable of coordinating movement and hunting strategies. The success of dinosaurs was closely tied to environmental conditions. The Mesozoic climate was generally warmer and more stable than preceding periods, with high levels of atmospheric carbon dioxide and minimal polar ice. This climate favored ectothermic metabolisms, allowing reptiles to thrive where mammalian competitors remained small and ecologically constrained. Vegetation was abundant and diverse, supporting both herbivorous and carnivorous lineages. Dinosaurs occupied nearly every ecological niche, from forest floors to riverbanks, demonstrating the versatility of their body plans. Physiological innovations contributed to their dominance. Hollow bones reduced weight without sacrificing structural integrity, facilitating both bipedal locomotion and, in some lineages, flight. Respiratory adaptations, including early forms of air sacs, enhanced oxygen delivery, supporting sustained activity in larger bodies. Teeth, claws, and digestive systems diversified in parallel with feeding strategies, allowing precise exploitation of available resources. Reproductive strategies, including egg-laying with protective nesting behaviors, ensured the persistence of populations even in fluctuating environments. Dinosaurs were not uniform in form or behavior. Saurischians, including theropods and sauropodomorphs, evolved bipedal predation and gigantism, respectively. Ornithischians explored armored forms, social behavior, and complex herbivory. Even within lineages, adaptive experimentation persisted. Some theropods developed feathers, not initially for flight, but likely for thermoregulation, display, or sensory enhancement. These innovations, preserved in fossil impressions, illustrate that dinosaur evolution was a story of experimentation guided by ecological pressures and opportunities. The rise of birds further underscores the evolutionary continuity within the reptile lineage. Feathered theropods, such as Archaeopteryx and later Maniraptorans, exhibit an array of skeletal, muscular, and neurological traits, linking them directly to modern birds. Flight, however, did not emerge as a sudden leap. It was the culmination of incremental modifications, feather development, lightweight skeletons, forelimb mobility, and behavioral adaptation. Birds are living dinosaurs, a testament to the persistence of Mesozoic innovation into the present. Mesozoic ecosystems were structured by these ecological experiments. Predators and prey co-evolved in dynamic networks. Large herbivorous sauropods influenced vegetation structure, nutrient distribution, and soil formation. Predatory theropods regulated herbivore populations, creating feedback loops that maintained ecosystem balance. Insects, smaller reptiles, and early mammals occupied supporting roles, exploiting niches that dinosaurs left unoccupied or could not efficiently utilize. The Mesozoic biosphere was a complex web, orchestrated by the interactions of these dominant reptiles. Dinosaurs thrived where mammals did not due to a combination of physiology and ecology. Warm-bloodedness had not yet become widespread. Dinosaurs were able to exploit ambient temperatures to maintain activity while minimizing energy expenditure. Large body size reduced predation risk and allowed efficient consumption of abundant plant matter. Ectothermic efficiency, combined with structural innovation, provided resilience in diverse environments. Mammals, still small, nocturnal, and ecologically limited, remained largely peripheral during this era. Extinction, however, remained a constant threat. Reptilian dominance was contingent, not absolute. Volcanic activity, climate fluctuations, and ecological shifts had eliminated dominant lineages before. The Mesozoic, though stable in relative terms, was not immune. Adaptive radiation was ongoing, but the underlying vulnerability of all ecosystems, dependence on energy flow, climate stability, and reproductive success, persisted. Dinosaurs, for all their dominance, remained subject to the same rules that had shaped life since its inception. The Age of Reptiles demonstrates the principle that evolutionary success is context-dependent. Dominance arises not from intrinsic superiority, but from ecological fit, energetic efficiency, and adaptive flexibility. Dinosaurs illustrate the power of form and physiology to exploit opportunity, while also highlighting the fragility inherent in specialization and gigantism. Their reign was long and productive, yet poised on the edge of abrupt environmental change. Within tens of millions of years, Earth had become a Mesozoic world. Forests of conifers and ferns, seas rich with marine reptiles, skies patrolled by pterosaurs, and continents dominated by a spectacular diversity of dinosaurs. Life had expanded in size, complexity, and behavior far beyond any previous era, exploiting every available niche. Yet, even this apparent triumph would not last indefinitely. The systems that had allowed reptiles to thrive, climate, energy, and ecological balance, remained vulnerable to rapid, catastrophic change. Reptiles had mastered the Earth for more than 160 million years, evolving intricate forms, behaviors, and interactions. Yet dominance in the natural world is always provisional. The very mechanisms that allow a lineage to thrive, gigantism, specialization, and ecological interdependence, also make it susceptible to sudden disruption. The Age of Reptiles was both a story of unparalleled achievement and a prelude to the limits imposed by planetary forces, revealing that no rain, no matter how enduring, is permanent. For over 160 million years, dinosaurs dominated Earth. They had conquered every terrestrial niche, diversified into forms of staggering variety, and reshaped ecosystems on a global scale. Yet in an instant, measured not in millions but in hours and days, that reign came to an abrupt halt. Approximately 66 million years ago, a massive asteroid, 10 to 15 kilometers in diameter, collided with Earth near what is now the Yucatan Peninsula. The evidence is undeniable: a global layer of iridium-rich clay, shocked quartz, and the 180-kilometer-wide Chicxulub crater, geological fingerprints of a catastrophe unprecedented in human experience. The impact released energy equivalent to billions of nuclear bombs detonating simultaneously. The immediate effects were catastrophic. A supersonic shockwave rippled through the crust and oceans, leveling forests, vaporizing organisms near the impact site, and triggering massive earthquakes and tsunamis across the globe. Material ejected into the atmosphere rained down as molten debris, igniting widespread firestorms that consumed vegetation and produced smoke so dense it shrouded the planet in darkness. For months, perhaps years, sunlight was drastically reduced. Photosynthesis halted. Global temperatures initially spiked from thermal radiation and then plummeted as particulate matter blocked incoming light. Ecosystems collapsed. Food chains that had persisted for millions of years disintegrated. Large-bodied animals, particularly dinosaurs, were disproportionately affected. Their energetic requirements made survival in a disrupted, resource-scarce world nearly impossible. Carnivores starved as herbivores disappeared, and entire lineages vanished within geological moments. Yet extinction was selective. Small-bodied, adaptable organisms fared better. Creatures capable of burrowing, hibernating, or subsisting on detritus had a survival advantage. Early mammals, already nocturnal and mostly small, were able to endure the collapse of the Mesozoic ecosystems. Birds, surviving lineages of theropods, persisted in part because of their metabolic flexibility, mobility, and dietary generalism. Even some plants survived in refugia, spreading rapidly in the aftermath to stabilize recovering ecosystems. The asteroid event demonstrates the role of contingency in evolutionary history. Dinosaurs had thrived for tens of millions of years, shaped by adaptation and selective pressures. Yet survival was not guaranteed by complexity, dominance, or size. A chance cosmic collision, entirely independent of the biological hierarchy, reset the ecological landscape. Evolutionary success, no matter how long or spectacular, can be undone by forces that are fundamentally random and external to life itself. In the wake of the disaster, ecosystems were drastically simplified. For millions of years, recovery was slow, constrained by energy availability, climatic instability, and the extinction of apex predators. Early mammals, small reptiles, birds, amphibians, and insects exploited the newly vacant niches. The absence of large competitors created evolutionary opportunity on a scale unseen since the Permian-Triassic recovery. Extinction had cleared the stage, leaving space for lineages previously suppressed to diversify and expand. Evidence of the impact's magnitude is preserved in rock layers worldwide. Iridium anomalies mark the thin sediment deposited during the event, while spherules of glass created from vaporized rock testify to molten debris raining down from the sky. Fossil records show abrupt disappearances of numerous species, from marine plankton to terrestrial vertebrates. Sedimentary deposits indicate massive tsunamis and wildfires. Geochemistry reveals abrupt shifts in carbon isotopes, reflecting ecosystem collapse and rapid carbon cycle disruption. Together, these lines of evidence form one of the most compelling and well-studied extinction events in Earth's history, a benchmark for understanding the interplay between chance, catastrophe, and evolutionary trajectory. This catastrophe did not merely end an era, it reshaped the rules of life. With the large reptiles removed, ecological hierarchies were reset. The surviving mammals, birds, and other small lineages were poised to radiate, exploiting resources and environments previously inaccessible. Evolutionary pressures favored flexibility, rapid reproduction, and small body size, traits that had been secondary during the age of reptiles, but now became primary drivers of success. The extinction event illustrates a sobering principle. Dominance is never permanent, and survival is contingent on traits that may have been marginal under previous regimes. The fate of dinosaurs was not due to biological failure, but to vulnerability in a world governed by both biological and astronomical forces. Life's trajectory is subject to the interplay of adaptation, opportunity, and randomness, a complex system in which even the most spectacular lineages can vanish almost overnight. In the shadows of the devastated ecosystems, lineages that had lived in marginal habitats now became central actors. Mammals, once small and nocturnal, were freed from predation and competition. Birds, descended from theropods, could exploit niches in trees, air, and on the ground. Insects, amphibians, and resilient plant species expanded into newly available habitats. The surviving biosphere was smaller, simpler, and vulnerable, yet filled with potential for diversification. The asteroid impact marked the end of the Mesozoic and the beginning of a new chapter in life's history. From the ashes of extinction, evolution would experiment anew, producing unprecedented forms, body plans, and ecological interactions. The mechanisms that had driven the rise of reptiles, energy efficiency, structural adaptation, and ecological dominance were suddenly subordinated to traits favoring flexibility, resilience, and rapid response. The world had burned, and in doing so, it had prepared the stage for the emergence of new giants, mammals and birds, that would dominate the ensuing Cenozoic era. After the extinction of the non-avian dinosaurs, roughly 66 million years ago, Earth's ecosystems were left in profound disarray. The towering archosaurs, the apex predators and giant herbivores of the Mesozoic, were gone. Forests, floodplains, and coastal environments lay in flux. This catastrophic reset created a vacuum, ecological space in which small, adaptable organisms could expand. Among them were mammals, long present but suppressed during the Mesozoic, largely nocturnal, small-bodied, and ecologically constrained. Freed from the shadow of dinosaur dominance, mammals began a remarkable evolutionary radiation, one that would culminate in forms capable of intelligence, endurance, and, eventually, planetary modification. The earliest Cenozoic mammals were tiny, generalized creatures, often weighing only tens to hundreds of grams. Fossil evidence from the Paleocene indicates insectivorous and omnivorous habits, reflecting both dietary flexibility and opportunistic survival strategies. These animals possessed relatively high metabolic rates for their size, endothermic physiology that permitted nocturnal activity, and heterodont dentition, allowing the processing of diverse foods. Small size and reproductive efficiency were critical to persistence in an unstable post-extinction world. Within a few million years, mammalian diversity expanded dramatically. The Eocene, beginning roughly 56 million years ago, saw the proliferation of numerous lineages. Early ungulates, primates, rodents, carnivores, and proboscideans emerged. Fossil evidence shows increasing specialization in locomotion, feeding, and sensory systems. Limb morphology diversified to accommodate cursorial running, climbing, digging, and swimming. Dentition adapted for specific diets, from folivory to carnivory, reflecting selective pressures imposed by available resources and ecological niches. Metabolic and thermoregulatory adaptations were crucial. Mammals maintained high basal metabolic rates and endothermy, allowing activity independent of ambient temperature. This physiological flexibility permitted colonization of environments previously inhospitable to reptiles of similar size. Fur or hair provided insulation, while sweat glands and panting mechanisms in certain lineages allowed the regulation of heat in tropical or open environments. Such adaptations were central to survival in climates ranging from subtropical forests to temperate grasslands. The evolution of sociality accompanied ecological diversification. Fossil and comparative evidence suggest that certain mammals, particularly early primates, evolved behaviors that enhanced group cohesion, resource acquisition, and predator avoidance. Social structures permitted cooperative foraging, shared vigilance, and, in some lineages, parental investment beyond immediate reproduction. Neural expansion accompanied these behaviors, as the demands of coordination, communication, and memory imposed selective pressure on brain size and complexity. Primates, emerging in the Paleogene, provide a clear example of environmental pressures shaping cognition. Early primates were small, arboreal, and visually oriented. Forward-facing eyes provided stereoscopic vision, critical for navigating complex three-dimensional environments. Limb morphology evolved to permit grasping, brachiation, and manipulative behaviors. Fossil evidence and comparative neuroanatomy indicate relative expansion of the neocortex and temporal lobes, suggesting increased processing of spatial information, memory, and social cues. These neurological developments set the stage for later intelligence and behavioral flexibility. Dietary innovation further influenced cognitive evolution. Frugivory and omnivory required memory of resource locations, recognition of seasonal availability, and problem-solving for extraction. Early primates and other mammals faced fluctuating climates during the Eocene and Oligocene, requiring adaptability and foraging strategies. These environmental pressures favored individuals with enhanced learning, innovation, and social coordination, traits that would accumulate over millions of years into forms capable of tool use, cooperative hunting, and culture in select lineages. The diversification of mammals was also shaped by changing climates and continental configurations. The Paleocene and Eocene were characterized by globally warm temperatures, high atmospheric CO2, and relatively low polar ice. Forests expanded across high latitudes, facilitating arboreal niches. As the Cenozoic progressed, climate cooled, grasslands emerged, and ice sheets developed in the Oligocene and Miocene. These shifts created selective pressures favoring endurance, migratory behavior, and dietary flexibility. Cursorial adaptations in ungulates, long-limbed predators, and social herding behavior all reflect responses to changing vegetation and predation patterns. Evolutionary experimentation during this era was not uniform. Extinction pulses, such as those associated with the Eocene to Oligocene transition, acted as bottlenecks, favoring lineages that could survive climatic volatility. Large-bodied mammals often became more vulnerable during these intervals, while small, adaptable species persisted and diversified. These episodes reinforced the principle that evolutionary success depends on flexibility, efficiency, and reproductive resilience rather than inherent dominance. Neurological evolution among mammals was remarkable. Encephalization, the relative expansion of brain mass compared to body size, occurred independently in multiple lineages. Primates, cetaceans, and certain carnivores exhibit evidence of enhanced cortical development associated with sensory processing, spatial reasoning, and social behavior. Fossil endocranial casts and comparative anatomy indicate that these expansions coincided with ecological complexity, foraging challenges, and social interaction. Intelligence emerges not as a goal, but as a consequence of environmental demands, energy availability, and behavioral complexity. Among mammals, the primate lineage ultimately set the conditions for the emergence of hominins. Early primates displayed grasping hands, opposable digits, stereoscopic vision, and flexible joints. These traits, in combination with dietary and social pressures, created selective landscapes in which tool use, problem-solving, and learning could provide adaptive advantages. Fossil evidence from the Miocene shows hominoid ancestors exploiting diverse habitats, from dense forests to open woodlands, responding to fluctuating climates and competition. Cognitive and manipulative abilities were gradually selected, refining neural circuits and muscular coordination necessary for later human behavior. Simultaneously, mammals exploited every available ecological niche. Aquatic lineages, such as early whales and sirenians, transitioned back into water, demonstrating physiological innovation in thermoregulation, respiration, and locomotion. Flying mammals, namely bats, evolved powered flight and echolocation, exploiting nocturnal aerial niches. Herbivorous and carnivorous mammals diversified in body size, dental morphology, and digestive specialization, illustrating the principle that mammalian evolution is shaped by energetic constraints, environmental opportunity, and ecological interaction rather than arbitrary superiority. By the late Miocene and Pliocene, Ecosystems had stabilized into structures familiar in many ways today: grasslands, forests, wetlands, and coastal plains. Mammals dominated most terrestrial environments, while birds, reptiles, and insects occupied complementary roles. Within this context, the hominin lineage emerged, small-brained at first but exhibiting bipedal locomotion, hand dexterity, and social coordination. These traits were selected under pressures of environmental fluctuation, resource scarcity, and predation. Intelligence and tool use were not inevitable, but became advantageous responses to ecological complexity. The radiation of mammals demonstrates the interplay between chance, adaptation, and contingency. Post-dinosaur ecological space created opportunity, physiological flexibility, endothermy, and reproductive efficiency allowed survival. Behavioral complexity, social structures, and cognitive expansion permitted exploitation of diverse niches. Extinction events, climate fluctuations, and environmental variability acted as selective filters, shaping lineages toward flexibility, problem-solving, and endurance. Evolution was neither linear nor directed. It was opportunistic, contingent, and constrained by energy, morphology, and ecological interaction. From tiny survivors of a catastrophic extinction, mammals transformed the planet over tens of millions of years. They evolved size, intelligence, social complexity, and ecological dominance. Primates, as a subset, developed manipulative hands, stereoscopic vision, and cognitive capabilities suited for environmental problem-solving. These innovations prepared the stage for a lineage that would, for the first time, reshape the world not only through adaptation, but through culture, technology, and environmental engineering. Mammalian evolution demonstrates that intelligence, endurance, and social complexity are products of selective pressures, not inevitabilities. Traits that facilitate survival under fluctuating climates, predation pressures, and resource scarcity accumulate over deep time, producing organisms capable of unprecedented ecological impact. From these foundations, one lineage would eventually emerge to transform Earth once more, altering not only ecosystems, but the very trajectory of planetary processes. From the small nocturnal mammals of the Paleocene, evolution had sculpted an array of forms capable of surviving, diversifying, and experimenting across the planet. Among these, the primates had developed an exceptional combination of traits: dexterous hands, stereoscopic vision, social complexity, and large brains relative to body size. These features, honed over millions of years, created the conditions for one lineage to diverge toward unprecedented cognitive capabilities. The emergence of Homo sapiens was neither inevitable nor miraculous, but the result of gradual, cumulative evolutionary pressures acting over hundreds of thousands of generations. The human lineage diverged from other great apes roughly 6 to 7 million years ago in the midst of the Miocene and Pliocene epochs. Fossil evidence combined with molecular phylogenetics suggests a complex pattern of branching and hybridization, with multiple hominin species coexisting, interacting, and sometimes interbreeding. Australopithecines, small-bodied bipedal primates, displayed adaptations for both arboreal and terrestrial locomotion. Their skeletal structure indicates that upright walking evolved to facilitate energy-efficient travel across expanding savannas, while retaining hands capable of manipulation and climbing. By roughly 2.5 million years ago, early members of the genus Homo appeared, including Homo habilis and Homo erectus. Stone tools, evidence of butchery, and increasingly sophisticated behaviors mark this period. The archaeological record shows incremental improvements in tool complexity, from simple flakes to hand axes and eventually composite tools. Such innovations reflect selective pressures favoring cognitive flexibility, problem-solving, and social learning. Homo erectus demonstrates another crucial milestone: the ability to control fire. Fire provided warmth, protection, and the means to cook food, dramatically increasing energy availability. Cooked food allows for greater caloric absorption, supporting larger brain sizes and more energetically demanding cognition. Brain expansion was the central hallmark of hominin evolution. Endocranial volume increased from roughly 400 to 500 cubic centimeters in early Homo habilis to over 1,300 cubic centimeters in anatomically modern Homo sapiens. Neuroanatomical studies show disproportionate enlargement of the prefrontal cortex, temporal lobes, and parietal regions, areas critical for abstract thought, planning, social cognition, and language. These expansions were energetically expensive, demanding both dietary resources and adaptations in maternal care, life history, and social structures. Extended childhood and cooperative rearing may have facilitated neural development while providing selective advantages in learning and cultural transmission. The fossil record indicates that Homo sapiens emerged approximately 300,000 years ago in Africa, evidenced by specimens such as those from Jebel Irhoud in Morocco. Genetic studies support this African origin, with subsequent dispersals into Europe, Asia, and eventually the Americas. During this period, other hominin species, including Neanderthals, Denisovans, and Homo floresiensis, coexisted, sometimes exchanging genes with early Homo sapiens. This mosaic of interaction demonstrates that human evolution was not linear, but reticulated, shaped by migration, isolation, hybridization, and local adaptation. Cognitive sophistication reached a new level around 70,000 years ago, a period sometimes referred to as the cognitive revolution. Humans began to create symbolic art, complex language, and abstract social structures. Archaeological evidence, cave paintings, ornamental artifacts, and burial practices indicates the emergence of culture as a distinct evolutionary force. Culture allowed humans to accumulate knowledge across generations, transmit technological innovations, and coordinate large social groups, dramatically increasing adaptability and resilience. Symbolic thought created an internal feedback loop, the capacity to reflect on the environment, imagine alternatives, and plan collectively accelerated evolutionary and cultural experimentation. Agriculture, beginning around 12,000 years ago, marked a transformative shift. Humans moved from foraging to domestication of plants and animals, altering energy flows and nutrient cycles on regional and eventually global scales. The Neolithic revolution created population densities, social hierarchies, and settlements that would ultimately lead to cities and civilizations. Agriculture enabled unprecedented specialization, labor division, and technological development, but also introduced vulnerability, reliance on a limited number of species, susceptibility to environmental fluctuation, and the spread of disease in dense populations. The Industrial Revolution, beginning in the 18th century, amplified human influence to an entirely new magnitude. Fossil fuel combustion, mechanization, and technological innovation transformed energy availability, climate, and ecosystems. Humans became a dominant geophysical force, capable of altering atmospheric composition, hydrological cycles, and biodiversity. The concept of the Anthropocene arises precisely from this reality. Humans, through culture and technology, can modify planetary processes at scales comparable to natural geological forces. Evidence of this impact is global, from elevated greenhouse gases to plastic deposition, from mass extinctions to altered nutrient cycles. From a scientific perspective, the emergence of Homo sapiens illustrates several principles. Intelligence is an adaptive response to environmental variability and social complexity, not an end in itself. Cultural transmission allows selective pressures to operate through knowledge accumulation rather than solely through genetic evolution. Technological innovation creates feedback loops that accelerate change, decoupling adaptive success from purely biological processes. Human evolution is thus a combination of classical natural selection, cultural selection, and niche construction, the ability to modify one's environment to enhance survival. Genomic studies reveal additional layers of complexity. Humans exhibit high heterozygosity, indicating past interbreeding with other hominins. Epigenetic mechanisms suggest plasticity in gene expression in response to environmental and social stimuli, enhancing adaptive capacity. Comparative genomics shows that humans share approximately 98 to 99% of DNA with chimpanzees, yet small regulatory changes, particularly in neural development and gene expression timing, account for profound differences in cognition, social behavior, and culture. The evolutionary trajectory of Homo sapiens is now inseparable from its technological and cultural capacities. Agriculture, urbanization, and industry exemplify a feedback loop in which behavioral innovation alters selection pressures. Climate change, habitat transformation, and the global redistribution of resources are no longer incidental byproducts of natural processes. They are a direct consequence of human agency. Life, for the first time, is reflecting on itself, modifying its own trajectory consciously rather than passively. Speculative scientific hypotheses about the future of life consider several possibilities. One avenue is continued human-driven evolution. Artificial selection, genetic engineering, and synthetic biology may produce organisms, including humans, with novel traits tailored to specific environments or purposes. Another scenario involves ecological collapse or selective bottlenecks induced by climate change, disease, or technological hazards, favoring resilient lineages over complexity or intelligence. Astrobiological and evolutionary theory suggests that technological species may either persist and modify planetary systems or inadvertently accelerate extinction events, a pattern potentially observable on other planets. Some theorists propose that the next phase of evolution may involve convergence between biological and artificial systems. Humans may integrate technology at cellular, neural, or societal levels, producing entities that transcend traditional evolutionary constraints. In this view, life becomes a hybrid system, information-driven, networked, and globally interconnected. Evolutionary pressure may increasingly favor adaptability to engineered environments, resilience to anthropogenic hazards, and capacity for self-modification. Alternatively, evolutionary trajectories could be shaped by stochastic processes. Even in a technologically advanced species, random events, asteroid impacts, supervolcanic eruptions, or pandemics could reset selective landscapes, favoring traits unrelated to intelligence or technological sophistication. Extinction remains a constant, reminding us that dominance is provisional and survival is contingent on adaptability, not predetermination. Thus, the emergence of humans completes a long arc, from single-celled replicators through microbial innovation, multicellularity, ecological experimentation, and mammalian intelligence, to a species capable of reflecting on its own role in planetary systems. Homo sapiens exemplifies both continuity and novelty, a product of deep evolutionary history, yet a catalyst for change at a scale unprecedented in Earth's history. Our species illustrates that intelligence and culture are not ends in themselves, but adaptive responses to environmental complexity, energy availability, and social networks. As we consider the future, humanity occupies a unique position. For the first time, life can intentionally modify its trajectory, accelerating adaptation through knowledge, technology, and global coordination. Yet, with this power comes responsibility. Ecological stewardship, climate mitigation, and the preservation of biodiversity are not moral luxuries but requirements for continued existence. Evolution has produced a species capable of reshaping not only its own destiny, but that of every other organism on the planet. After 4.5 billion years of blind experimentation, the story of life may continue in multiple ways. One possibility is the ongoing evolution of Homo sapiens itself, producing physiological, cognitive, and technological novelties beyond current imagination. Another is the emergence of entirely synthetic life forms, either genetically engineered or artificially constructed, that may rival or surpass humans in adaptability. A third is the natural selection of species resilient to anthropogenic pressures, producing a post-human biosphere that bears the imprint of past catastrophes, climate shifts, and human engineering. In every scenario, the central lesson of deep time remains. Life is persistent, innovative, and contingent. From the first replicators to microbial dominions, the oxygenation of the planet, the rise and fall of reptiles, the mammalian radiation, and the emergence of humans, life has continuously experimented, failed, adapted, and expanded. Humanity is not the conclusion of this experiment, only the most recent actor. What comes next is unknown, yet the principles are clear. Adaptation, innovation, contingency, and energy constraints will continue to shape life, whether mediated by human intelligence, natural processes, or artificial creation. Life has at last become self-conscious. It observes its own history, manipulates its own evolution, and modifies its own environment. Whether this will lead to enduring planetary stewardship, novel forms of intelligence, or eventual collapse is uncertain. What is certain is that the next chapter, like all chapters in the 4.5 billion-year story of life, will emerge from the interplay of chance, adaptation, and energy. After billions of years of blind experimentation, life stands poised to decide, in its own complex and uncertain way, what it will become next. If this journey through our past helped you understand what it takes to survive, subscribe to Science of Survival. We release new documentaries every week, exploring extinction, adaptation, and the fragile line that keeps humanity alive.